But how do we make more? Okay, so let's talk about you. You are made of cells, trillions of them. Like an absolutely mind-boggling number of tiny biological building blocks, all working together to make you, well, you, your skin, your brain, your eyeballs, your toenails. And these cells don't just sit there. They are constantly working, they get old, they get damaged, your body needs more of them to grow. So, your body has to make new cells. This process is called cell division. And it's happening inside you right now, millions of times a minute. It's one of the most fundamental processes of life itself. Without it, nothing could grow, heal, or even exist in the first place. Now, your body has two main ways of doing this cell division thing. The first, and most common, is called mitosis. Think of mitosis as the body's copy machine. One parent cell becomes two identical daughter cells. Same genetic information, same number of chromosomes, same everything. This is perfect for when you need to grow taller or heal a paper cut. It's a reliable, straightforward process that keeps your body running smoothly day in and day out. But what about making a whole new person? A new plant? A new fungus? If you just used mitosis for reproduction, the baby would end up with twice as much DNA as its parents. Then its kids would have 4 times as much, and so on. Pretty soon, you'd have cells bursting with way too much genetic material. A total disaster. So nature needed a different strategy. It needed a way to cut the amount of genetic information in half, specifically for the cells involved in sexual reproduction. This is where our star player for today comes in. This special type of cell division is called meiosis. Meiosis is not about making identical copies, oh no, it's about creating variety. It's about shuffling the genetic deck and dealing a new hand. Instead of making 2 identical cells, meiosis takes 1 cell and, through a much more complex and fascinating 2-part process, creates 4 unique cells, each with only half the number of chromosomes as the original parent cell. These special cells are what we call gametes, sperm in males' eggs in females. And this process is the very reason why, unless you have an identical twin, there is no one else on Earth quite like you. So we've established that meiosis is the specialist. While mitosis is the general contractor building the house, meiosis is the artist creating a unique masterpiece. The main purpose of meiosis is twofold. Both parts are incredibly important for the continuation of life. First, reducing the chromosome number. Most of your body's cells, called somatic cells, are diploid. That means 2 sets of chromosomes. Before a cell can dive into the complex dance of meiosis, it has to prepare. This preparation stage is called interphase. It's pretty much identical to the interface that happens before mitosis. You can think of it as the cell's training montage. It's bulking up, stocking up on supplies, and most importantly, making a complete copy of its DNA. The cell is not technically dividing during interphase, but it's an incredibly active and crucial period. It's doing all the behind-the-scenes work to ensure the main event goes off without a hitch. This phase is divided into 3 parts, G1, S, G2. The first part is the G1 phase, or gap 1. During this stage, the cell is just living its life, but it's also growing. Once the cell gets the green light, it moves into the S phase, which stands for synthesis. This is the main event of interphase. Here, the cell meticulously duplicates all of its DNA. Finally, the cell enters the G2 phase, or gap 2. The cell is poised to exit interphase and begin the dramatic and beautiful process of meiosis 1. Alright, the cell has finished its prep work in interphase, and now it's time for the main act, meiosis 1. The primary goal of meiosis 1 is to separate homologous chromosomes. Remember, homologous chromosomes are the pairs you have. One from mom, one from dad. They carry genes for the same traits, but not necessarily the same versions. Meiosis 1 is all about taking these pairs and separating them, so each of the 2 new cells gets 1 chromosome from each pair. This first meiotic division is called the reductional division. It reduces chromosome sets from 2, diploid, to 1, haploid. Each chromosome still has 2 sister chromatids, but the total number of chromosomes is halved. Meiosis 1 is continuous, but we break it into 4 phases. Prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 is the longest and most complex, with several substages. This is where genetic shuffling begins. Metaphase 1, chromosome pairs line up. Anaphase 1, they are pulled apart. Telophase 1, the cell begins to split in 2. Keep the big picture in mind. The goal is 2 haploid cells from 1 diploid cell. Without this reductional division, sexual reproduction would be impossible. Welcome to the most exciting and intricate phase of meiosis, prophase 1. If meiosis were a movie, this would be the action-packed, plot-twisting first act. So much happens here. First, just like in mitosis, the chromatin in the nucleus condenses and coils up, becoming visible as distinct chromosomes. The nuclear envelope starts to break down. Spindle fibers, like cellular ropes, begin to form. But then, something truly unique and amazing occurs. The homologous chromosomes, the one from your mom and the one from your dad, find each other and pair up. This pairing is called synapsis. Imagine all 46 of your chromosomes floating around. Each one has to find its specific partner out of the whole crowd. Chromosome 8 from your mom finds chromosome 8 from your dad. This precise pairing forms a bivalent, also called a tetrad. It consists of 4 chromatids all lined up together. 2 sister chromatids on the maternal chromosome, 2 sister chromatids on the paternal chromosome. Now for the coolest part, crossing over. Homologous chromosomes snuggle up in their tetrad and literally exchange genetic material. A piece of a maternal chromatid breaks off and swaps places with the matching paternal piece. These exchange points are called chiasmata. This is not a mistake, it's programmed and deliberate. The result? Brand new hybrid chromosomes, a mosaic from both parents. It's the ultimate genetic remix. After the incredible genetic shuffling of prophase 1, the cell moves into the next phases of meiosis 1, metaphase 1, anaphase 1. In metaphase 1, the drama moves to the center of the cell. The tetrads, those paired up homologous chromosomes, migrate and line up along the cell's equator, an imaginary line called the metaphase plate. The spindle fibers that formed earlier now attach to the centromeres of each chromosome, but with a crucial difference from mitosis. Spindle fibers from one pole attach to one homologous chromosome of the pair, and fibers from the opposite pole attach to the other. This alignment is completely random. For each of the 23 pairs, it's a 50-50 chance whether the maternal or paternal chromosome lines up on one side or the other. This is called independent assortment. Think about it like flipping 23 coins. The orientation of one pair has no influence on the orientation of any other pair. This randomness is another massive source of genetic variation. With 23 pairs of chromosomes, the number of possible combinations of maternal and paternal chromosomes that can be sorted into the daughter cells is 2 to the power of 23, which is over 8 million. Next up is anaphase 1. The ana prefix means up or apart, and that's exactly what happens. The spindle fibers contract, pulling the homologous chromosomes away from each other toward opposite poles of the cell. It's a cellular tug-of-war. But, importantly, the sister chromatids remain attached at their centromeres. It's the pairs that are separating, not the individual chromatids. We've made it through the first division and now have 2 haploid cells, but we're not done. Each chromosome in these cells is still made of 2 sister chromatids. The goal is to create gametes with single unduplicated chromosomes, so we need another round of division. This is meiosis 2. If you understand mitosis, you already understand meiosis 2. It is essentially a mitotic division, but it happens in 2 haploid cells instead of 1 diploid cell. That's why it's often called the equational division, because it doesn't reduce the chromosome number, it just separates the sister chromatids. Between meiosis 1 and meiosis 2, there may be a brief resting period called interkinesis, but it's not the same as interphase. Critically, there is no S phase. The DNA does not replicate again. The cells already have the duplicated chromosomes they need, so they can jump right into the second division. Meiosis 2 is divided into 4 phases: prophase 2, metaphase 2, anaphase 2, telophase 2. These phases happen simultaneously in both haploid cells created at the end of meiosis 1. The final result? 4 haploid cells, 4 gametes. We have reached the very end of the line. Telophase 2 has set up the 4 new nuclei. All that's left is to divide the cytoplasm to create the final separate cells. This process, called cytokinesis, happens concurrently with telophase 2. Cell membranes begin to pinch inward, forming a cleavage furrow. The furrow deepens until it splits each cell completely in 2. The result of this journey? 4 brand new haploid cells. Each of these 4 cells is genetically unique. There's an interesting twist in animals. In males, division is equal. Cytoplasm splits evenly. Spermatogenesis yields 4 functional sperm of similar size. Each sperm is a lean swimming machine. It's an efficient process making massive numbers of gametes. A healthy male produces millions of sperm daily. In females, it's very different. Oogenesis shows wildly unequal cytoplasm division. During meiosis 1 and 2, almost all the nutrient-rich cytoplasm organelle's cellular machinery goes to just one daughter cell. The other cells, polar bodies, are tiny and get almost nothing. They're little packages of discarded chromosomes and are not viable. They degenerate. End result. Not 4 functional cells, but one large, well-stocked egg, ovum, and 2 or 3 tiny polar bodies. Why the inequality? The egg has a big job. If fertilized, it needs resources, nutrients, mitochondria molecular instructions, to sustain the embryo for the first few days before implantation. Fascinating fact. The mitochondria in your cells, which provide your energy, were inherited from your mother, from the cytoplasm of her egg. The importance of meiosis cannot be overstated. It is one of the pillars of biology, fundamental to the life cycles of nearly all complex multicellular organisms on Earth. It is, quite literally, the spice of life.